Hello, my name is Rojiro and I am gonna explain the parts and the function of a leaf. So first, looking at the picture or the drawing, we have at the top we have the stoma and the guard cells. Stoma allow a plant to take in carbon dioxide that is needed for photosynthesis. Guard cells are cells surrounding each stoma. They help regulate the rate of transpiration by opening and closing the stomata. Next, we have the waxy cuticle. Waxy cuticle are made up of hydrocarbons and wax. This makes the waxy cuticle hydrophobic or repels water. Just underneath the waxy cuticle, we have the upper epidermis. The cells of the upper epidermis are very closely packed together. The upper epidermis has mostly protective function. It allows light to come in and it also prevents water loss. Under the upper epidermis, we have the palisade mesophyll. The palisade mesophyll consists of two layers of closely packed, long cylindrical cells. The palisade mesophyll also contains numerous of chloroplasts. This makes the palisade mesophyll the main site of photosynthesis. Under the palisade mesophyll, we have the spongy mesophyll. The shape of the spongy mesophyll are irregular in shape. They contain some chloroplasts. This means that they can do photosynthesis, but not as much as the palisade mesophyll. The cells of the spongy mesophyll are also pretty far apart, and the area between them is called the intercellular space. Next, we have the vascular bundle. The vascular bundle is round and responsible for moving products around leaf. The outside coating of vascular bundle is made up of large group of closely packed cells. These cells are called bundle sheath cells, and they keep the vascular bundle together. Inside the bundle sheath is the tightly packed tubules called xylem and phloem. The function of xylem is to bring water up from the roots of the plants and distribute it to the stem and the leaves. The function of phloem is to bring glucose to all of the plant and distribute to all of the plants. Just under the vascular bundle and the spongy mesophyll, we have the lower epidermis. Just like the upper epidermis, the lower epidermis has cells that are packed very closely to prevent water loss. Finally, under the lower epidermis, we have one more waxy cuticle. Just like on the top, it helps to prevent water loss.
Moving on to the mechanism of stomata, I will recall the functions of stomata and guard cells, which are two parts that is important in this mechanism, and I will recall it because it was explained by Ranjiro at the internal parts of the plants, at plant's leaf. So the function of stomata, two main functions of stomata are to allow for the uptake of carbon dioxide and to limit the loss of water due to evaporation. And in where stomata is, it usually is protected by the guard cells, which its main function is to help regulate the rate of transpiration by opening and closing the stomata. Now, moving on to the mechanism of stomata. When the guard cells are turgid or swollen, the stomata opening will be large. And when the guard cells are losing water or becoming flaccid, the stomatal opening will be closed and it will be much smaller. Then, in what cases the stomatal opening is open and closed? So, usually when the stomata closes, the potassium ions move out from the cells. The water moves out from the vacuoles. The guard cells shrink in terms of size, and water potential of the guard cells increase, in which, if the stomata opens, the potassium ions enters the cells. Water moves into the vacuoles. The guard cell expands and the water potentials of the guard cells drop. So I will show the picture of the open stomata and the closed stomata. So as you can see, this is the open stomata, and usually when the open stomata, the H2O, which is the water, the potassium ions usually moves inside to the stomata, and at the closed stomata part, the water and the potassium ion moves out from the stomata. Okay.
Photosynthesis. Photosynthesis is a chemical reaction that takes place inside a plant, producing food for the plant to survive. Carbon dioxide, water, and light are all needed for photosynthesis to take place. Photosynthesis mostly happens in the leaf of a plant. Origin of the word. Photosynthesis derived from the Greek word photo or phos, which means light, synthesis or synthesis, which means to put together. Photosynthesis is the process used by the plant and other autotrophic organisms to convert light energy or sun to chemical energy that can later be released to fuel the organism's activities. Raw materials. Carbon dioxide. Carbon dioxide is the raw materials used for the formation of glucose. Plant obtain it from the atmosphere through stomata. Water. Water is the raw materials where the plant absorb it from the soil with the help of root system. Other materials like nitrogen, phosphorus, iron, and magnesium are also taken up from the soil. Solar energy is the raw materials where the solar energy is trapped by the chlorophyll. This is present inside the chloroplast.
There are two stages of photosynthesis. The first one is light-dependent reaction stage, and the second one is light-independent reaction stage. So the first one is light-dependent reaction stage. I'm going to discuss it. It uses light energy and water to produce ATP, NADPH, and oxygen. So how does it work? Firstly, light energy causes excitation of electrons in the photosystem 2. So what is photosystem? Photosystem is large complex of proteins and pigments that are optimized to harvest light and plays a key role in the light reactions. So there are two types of photosystems. The first one is photosystem 1 that has pigment P700, and the second one is photosystem 2 that has pigment P680. So light energy causes excitation in the photosystem 2, and the P680 loses an excited electron. That excited electron then releases energy, then goes to the lower state of energy. Some of the energy released is used to pump the hydrogen ions from the stroma to the thylakoid lumen, increasing the gradient of hydrogen ions in the thylakoid lumen. After the excitation, the P680 loses an electron and it becomes P680+, so it needs to attract an electron to become more stable. The water or H2O is oxidized by the P680+, so it is fallen apart into oxygen and hydrogen ions. The hydrogen ions also help to increase the gradient of the hydrogen ions in the thylakoid lumen. So the high gradient of hydrogen ions in the thylakoid lumen causes the hydrogen ions to go back out through the ATP synthase. It helps to make the inorganic phosphate plus ADP into ATP, so it produces ATP. How about the electron that is excited before? It already went into lower and lower state of energy.
into the photosystem 1. So the photosystem 1 also receives light energy and experiences excitation. So the P700 also loses an excited electron. So it gains an electron from the photosystem 2 again. So what about the excited electron from P700? It goes into the lower state of energy. It converts NADP plus into NADPH. So the light dependent reaction has three byproducts. The first one is oxygen that is produced during the oxidation of H2O by the P680 plus. The two other products are ATP and NADPH, which are then used in the light-independent reaction stage.
So, continuing Steven's video, right now I'm going to discuss about the light-independent reaction stage. Light-independent reactions take place in plants, chloroplasts. In this process, sugar are made from carbon dioxide. The process, known as Calvin cycle, uses product from the light-dependent reaction, such as ATP and NADPH, and various enzymes. Therefore, light-independent reaction cannot happen without light-dependent reaction. Sugars made in the light-independent reaction are then moved around the plant by translocation. Takes place in the grana region of chloroplast. It is also anaerobic. Next, Calvin cycle. Like what I said before, the cycle that are happening in the light-independent reaction stage is called the Calvin cycle. So what's Calvin cycle? Calvin cycle refers to the light-independent reaction in photosynthesis that takes place in three key steps. First is fixation, second is reduction, and last regeneration. I'll discuss it further later on. Although the Calvin cycle is not directly dependent on light, it is indirectly dependent on light, since the necessary energy carriers like ATP and NADPH are products of light-dependent reactions. To form glucose, it takes 6 Calvin cycles to make a single glucose. Okay. So right now I'm going to discuss about the Calvin cycle. So this is how it looks like. So first, fixation, like what I say, is the first step or stage in Calvin cycle, which is here. In the stroma, in addition to the carbon dioxide, two other components are present to initiate the light-independent reaction. An enzyme called ribulose biphosphate carboxylase, or simply Rubisco, which is this, and three molecules of ribulose biphosphate, which is the RUBP. In this process, Rubisco catalyzes a reaction between the carbon dioxide and ribulose biphosphate, or we are going to use simply RUBP. For each carbon dioxide molecule that react with one RUBP, two molecules of 3-phosphoglyceric acid, or simply 3-PGA form, which is this. 3-PGA has 3 carbons and 1 phosphate. Each turn of the cycle involves only one RUBP and one carbon dioxide and forms 2 molecules of 3-PGA. Since there's 3 and there's 3, it can form 6 molecules of 3-PGA. The number of carbon atoms remain the same as the atoms move to form new bonds during the reaction. This process is called carbon fixation. Why? It is because the carbon dioxide is fixed from an inorganic form into organic molecules. The second stage is reduction, which is the reduction of 3-PGA. ATP and NADPH are used to convert the 6 molecules of 3-PGA into 6 molecules of a chemical called glycerol G-hide 3-phosphate, or G3P, which is from here, By the using of ATP and NADPH, we form this G3P. This is a reduction reaction because it involves the gain of electron by 3 PGA. Recall that a reduction is the gain of an electron by an atom or molecule. 6 molecules of both ATP and NADPH are used. We can see it right here. After they both give its electron, they'll go back to their form before, which is 6 ADP, and this one will split into 6 NADP plus and H plus. For ATP, energy is released with the loss of the terminal phosphate atom, converting it to ADP, no longer ATP. For NADPH, both energy and a hydrogen atom are lost, converting it to NADP+, which means that the hydrogen ion is going to be used. Both of these molecules return to the nearby light-dependent reactions to be reused and re-energized. So the third stage, the regeneration of RUBP. At this point, only one G3P molecule leaves the Calvin cycle and is sent to the cytoplasm and contribute to the formation of other compounds needed by the plant. Because the G3P exported from the chloroplast has 3 carbon atoms, it takes 3 turns of Calvin cycle to fix enough net carbon to export 1 G3P. Because each turn makes 2 G3Ps, thus 3 turns to make 6 G3Ps. 1 is exported while the remaining 5 G3P molecules remain in the cycle and are used to regenerate RUBP, which enables the system to prepare for more CO2 to be fixed. 3 more molecules of ATP are used in this regeneration reaction. As we can see here, it was supposed to have 6 G3P, but 1 molecule will be used and the rest 5 molecules of G3P will continue the Calvin cycle. Then, after many times of Calvin cycle, they will use the ATP, then get the electron back to the Calvin cycle. The ATP will no longer ATP but ADP, and they will form 3 molecules of RUBP. So as a recall, we'll go back to the first. As what my friend told, my teammate said, the internal parts of the leaf is somehow like this. We have the stoma, gourd cells, which is protecting the stoma, upper epidermis with waxy cuticle above it, palisade mesophyll below the upper epidermis for photosynthesis, spongy mesophyll for photosynthesis also, but not mainly occur. Then we have the vascular bundle, the lower epidermis, and another waxy cuticle below the lower epidermis. Next, the mechanism of stomata. Stomata is protected by the gourd cells, which its main function is to regulate the rate of transpiration by opening and closing the stomata. When the gourd cells are turgid, the stomatal opening will be large. When the gourd cells loses water, the stomatal opening will be closed. So this is the comparison where when stomata close and when stomata opens. Okay, next. Valeran also discussed what happened during stomata is open and what happened during stomata is closed. I'll recall, when stomata is opening, water or simply H2O and potassium ion or K plus will enter to the stomata. In closed stomata, water will exit and potassium ion will also follow exiting the stomata. Then photosynthesis is a chemical reaction that takes place inside a plant, producing food for the plant to survive. it needs carbon dioxide, water, and light, which is the raw materials. Then also our main topic for photosynthesis, we have the origin of the word photosynthesis, the raw materials for photosynthesis, and also the stages of photosynthesis, like what I and Steven discussed, which is the light dependent reaction stage and also light independent reaction stage. Origin of the word photosynthesis derives from the Greek word phos and synthesis. Phos means light, synthesis means to put together. Hence, we can say that photosynthesis is the process used by plants and other autotrophic organisms to convert light energy, which is from the sun, to chemical energy, which is the glucose, that can later be released to fuel the organism's activities. Raw materials, like what I said, carbon dioxide, water, and light. Light is also the solar energy because it's from the sun. Okay, and to the first one, light dependent reaction stage. Light dependent reaction stage is the use of energy to make two molecules needed for the next stage of photosynthesis. The next stage of photosynthesis is the light independent reaction stage. So in this light dependent reaction stage, it uses solar energy, or simply the sunlight, and carbon dioxide to form ATP and NADPH, and also oxygen. And later on in light independent reaction stage, the plant will use ATP and NADPH to make glucose. That's all from our group. We are group 4 X IPA. Thank you.